Mastering Biology 9700 4.1, the arrangement of cholesterol, glycolipids, and glycoproteins. Look, today we are skipping all the fluff. We're going to dive straight in and expose the one tiny fatal terminology mistake that costs students easy marks before they even start explaining the actual biology. In this explainer, we're looking directly at what the examiners actually want. We'll break down these complex concepts and build the exact analytical frameworks you need to hit those top mark bands. Let's get right into it. Cholesterol is in the membrane and helps it function. Okay, listen to that for a second. It's a frustratingly common student answer. And here are the actual stakes. Cambridge examiners will give you exactly 0 marks for that. 0. Why? Well, because the assessment framework for this topic prioritizes analytical depth over superficial breadth. You can't just state that something exists and does a job, right? Examiners require specific contextual details and logical, structured reasoning. An answer this vague is basically an immediate red flag that a candidate doesn't grasp the underlying theoretical foundations.
So, to fix this, we've got to master the core concepts with absolute precision. Let's look at our first key term, the glycolipid. This is simply a lipid with a carbohydrate attached, and it is absolutely crucial for cell recognition. You can think of it almost like an ID badge at a security checkpoint for the cell. Now, what's really interesting here isn't just the biological fact itself, it's what the principal examiner records tell us about it. They explicitly warn that candidates consistently confuse key terminology in this section. So, the golden rule straight from the examiners, you literally must define terms like glycolipid accurately before you even try to use them in your extended responses.
Let's see how this builds with our next component, the glycoprotein. This one is a protein with a carbohydrate attached, primarily serving as a receptor. Anchoring these solid biological facts is your direct remedy to those vague, 0-mark answers we just talked about. When you specifically identify a glycoprotein and define its structural nature, a protein plus a carbohydrate, you're showing the examiner that you genuinely grasp the foundational science rather than just taking a wild guess.
So how do all these pieces actually work together? Let's bring it into focus. Cholesterol regulates fluidity, glycolipids facilitate cell recognition, and glycoproteins act as receptors. But pay close attention to that final point, analytical depth and evidence-based reasoning. It is absolutely not enough to just memorize this list. You have to be able to apply these roles to completely novel scenarios. You've got to handle complex information to prove you understand exactly how they function within the dynamic, bustling environment of a living cell membrane.
Which brings us to a really critical turning point. In just a minute, I'm going to reveal the hidden examiner clue that changes your answer completely. It's a single detail that dictates exactly how you must frame your biological knowledge. But first, you need the procedural frameworks that make spotting that clue totally obvious. Because without understanding the methodology behind the biology, you'll honestly walk right past the clues the exam paper is practically trying to hand you. Welcome to the method mark strategy. In Cambridge 9700, marks are awarded for the actual process, not just the final answer. The mandatory statistical frameworks for analyzing membrane data map out exactly when to use which test. So, you're going to use the t-test for comparing means between two groups, you'll use chi-squared for analyzing categorical data, and you use the correlation test for finding relationships between variables. Look, if you apply the wrong statistical test to a dataset about, say, cholesterol concentration and membrane fluidity, your brilliant biological knowledge just isn't going to save your score.
And this ties directly into your practical experimental skills. Topic 4.1 isn't just theory sitting in a textbook, it heavily involves practical work. Standard memorization just won't cut it. You have to demonstrate that you can select the appropriate apparatus, follow sequential instructions, make highly accurate observations, record that data in suitable tables, and rigorously identify anomalies. It's all about extracting data from tables and graphs, interpreting those relationships, and calculating those relevant statistics we just talked about to draw solid evidence-based conclusions.
But even knowing the stats and the practical steps, students are still tripping up. And it really comes down to 3 massive traps. We dug deep into the Cambridge Principal Examiner report specifically for topic 4.1, and we found the top 3 shocking mistakes that are just dragging down candidate grades, series after series. If you can avoid these 3 pitfalls, you will instantly separate yourself from the average candidate.
All right, here are the examiner warnings in black and white. First up, answers are far too vague. Like we saw at the start, examiners want highly specific details. Second, students are constantly forgetting to check units and terminology. Leaving out a unit in a calculation or confusing a glycolipid with a glycoprotein, that's fatal for your score. Third, and honestly most importantly, answers are not specific to the given context. Having a perfect biological understanding is completely useless if your answer isn't tailored to the exact experimental context provided in the question.
To make sure you literally never make that third mistake, you've got to use this specific problem-solving framework that top-scoring candidates rely on. We call it deconstruct, solve, evaluate. First, identify the given information. Really anchor yourself in the context. Second, break the problem down into logical steps. Third, solve it systematically. Fourth, check for reasonableness. Like, ask yourself, does this actually align with the theoretical foundations of membrane fluidity? And finally, make justified recommendations. This framework forces you to actually use the context.
And that brings us straight to the evaluation stage. Top-scoring candidates don't just solve problems, they demonstrate real evaluative thinking. They question assumptions, they consider alternate perspectives, and they back up their decisions with hard evidence. When you're asked to evaluate the quality of evidence regarding membrane components, you absolutely must identify the limitations of the experiment. Spot the assumptions being made, consider alternate interpretations of the data, and explicitly state exactly how these factors affect the final conclusions.
Okay, let's put you in the hot seat for a second. This is an interactive challenge based directly on an authentic exam scenario. Ready? A student investigates the effect of temperature on membrane permeability. How do the components respond? Take a second. Seriously, pause and predict how you would structure this answer in your head. Are you visualizing the cholesterol? Are you thinking about the kinetic energy? Let's see how your predicted answer actually stacks up against what the examiners are desperately looking for.
Let's compare. A flawed answer sounds a bit like this. Heat makes cholesterol move and breaks the membrane. Again, way too vague. Now listen to a top-band answer. Increased kinetic energy disrupts cholesterol interactions, increasing fluidity and permeability. See the difference? This brilliantly illustrates our ultimate framework: concept to evidence to scientific reasoning. You start with the concept, kinetic energy. You provide the specific mechanism, disruption of cholesterol interactions, and you conclude with the scientific reasoning, increasing fluidity and permeability. That right there is how you secure top marks.
But here is the ultimate trap. That hidden clue I mentioned way earlier. You could write that perfectly crafted, top-band biological answer we just looked at, but what happens when the exam command word says evaluate instead of describe? I'll tell you exactly what happens. Misinterpreting that single command word can instantly cap your flawless biological explanation at the absolute lowest mark band. If you describe when you are actually supposed to evaluate limitations, all that precise terminology we just learned won't save you.
Command words dictate your entire strategy. So you've got to ask yourself, are you just memorizing biology, or are you actually learning how to take the exam? Understanding the science is only step one. Executing the exact required strategy is step two. To fix this, you need to watch our next explainer, which breaks down exactly how to decode these command words so you never ever leave marks on the table again. Subscribe to our YouTube channel, Ace the Exams, at Ace the Exams IGCSE to excel in your Cambridge examinations. We'll see you in the next one.